the theory of evolution by natural selection to our understanding of the planet and also of ourselves. So this is an event that's celebrated at institutions throughout the world, hundreds of places, and we're very fortunate to have this event here as well. And we're very, very fortunate to have Dr. Molly McDonough, who is from Chicago State University. And Dr. McDonough grew up in St. Paul, Minnesota, but moved to San Antonio, Texas in high school. She did her undergraduate at Texas State University in San Marcos, and where she studied wildlife biology. Then she went on to do a Master's of Science in San Angelo, Texas, with an emphasis, it was in biology, with an emphasis on molecular systematics of bats. And I'd forgotten this before you emailed me, but she described a new species of bat from Ecuador. Her accomplishments are such that we invited her, even though I'd forgotten that. She's done that. Then she went on to do her PhD at Texas Tech University, where she met one of our very new colleagues, Noé de la Sancha, from Environmental Sciences. And she continued her work on molecular systematics, but she changed her focus to African rodents. She did 3 postdocs, one at the National Museum of Natural History, where she studied the systematics of African rodents. She did another postdoc at the Center for Conservation Genomics, where she studied the evolution of pox viruses in African mammals. And then she finally did a Fulbright teaching postdoc at Masai Mara University in Kenya, which is where she went to interview from Kenya and Chicago State and got the job there, fortunately for us. And she's been at Chicago State University since 2018. So we're very happy to have you and looking forward to your presentation.
Thank you. Thank you so much for inviting me, and I love this celebration. And we do not celebrate it at Chicago State University, but I'm thinking you've inspired something, and we'll try and get a celebration going there as well. What I thought of doing today was I'll talk about how I use natural history collections to study the evolution of mammals. I'm going to talk about a specific skunk story today and their pathogens, and I thought this would be appropriate given Darwin Day, given his contribution of specimens to natural history collections and how those specimens have influenced the theory of evolution. As Windsor said, I did start off in Texas State, and I got my undergraduate degree in wildlife biology. The focus of that program was mainly management of game animals, though. And so I am sorry to say that I never really participated in undergraduate research while I was there. And so I'm sort of obsessed with, now that I'm a faculty member at Chicago State University, I always try and get students involved in undergraduate research. But I found myself always attracted to the non-game animals. And so for my wildlife management plan, it's like your kind of capstone. I was coming up with ways to manage populations of bats in Texas. And so they could see that I was different. They could see that I didn't fit in with that game management system. And toward the end of my degree, one of the faculty members reached out to me and asked if I'd be interested in conducting mammal surveys at a National Guard site. And so that was kind of my first experience working with mammals, learning how to survey them. And then I carried on doing a couple of seasonal jobs and wanted to apply for more permanent positions, but realized I needed a master's to do that. And so I was at a conference, and I met a woman there who is the leading bat biologist of Texas. And she's at a school, Angelo State University, that maybe you've never even heard of, but they have an amazing natural history collection. And so not only did I get experiences learning how to survey for bats, but she also taught me how to do molecular systematics. And so generating these phylogenies and looking at species boundaries. And like Windsor said, during this time, we got to describe a new species from the lowlands of Western Ecuador. But it was really this natural history collection that I spent most of my time learning how to prepare specimens, how to collect them in the field, and in such a way that they could be deposited for the long term. And really instilling this idea that these collections are going to be around for a very long time. And so we've got to come up with ways to keep them funded and protect them. And so I really had no plan to get a PhD, but as I was finishing up my master's and looking for jobs, I was recruited by one of the co-authors that was part of one of our manuscripts to pursue a PhD at Texas Tech University. And he offered a fellowship that would support me financially, and so I kind of couldn't resist. But I think it was a good decision in the end. My first semester there, I ended up going to Botswana to conduct a mammal survey. And I really got hooked on the African fauna. And so I built my dissertation around the ability to go back to Africa. And so my questions and hypotheses all revolved around going to these sites in Southern Africa. But I did do some science in addition to traveling. And I eventually got out of Texas. I mean, Texas is cool. It's a nice place. I mean, it's got all these different ecosystems and the rivers are amazing. But eventually we got out and we got to travel all over the place. And so my husband, Adam Ferguson, is my partner in crime for a lot of this stuff. And he's a collections manager of mammals over at the Field Museum. Part of the reason we ended up in Chicago. And we decided to put this together just to see all the places that we've either done field work or museum collections that we've visited. And then the darker ones are places we've lived for longer periods of time. And so as Windsor said, before I came to Chicago, I was teaching at the Masai Mara University in Kenya for a while. But then before that, you see this small little dot in Washington, DC. And that's where I did my postdoc. And part of my goal during postdoc was really to learn new technologies, and especially to learn how to get DNA out of really old and degraded stuff. And while I didn't work in this particular lab, they have a couple of these clean rooms there. I did learn how to get DNA out of old stuff and the methods, the specific methods involved in getting the least amount of contamination in these samples. One of the papers that was kind of instrumental in me sort of becoming obsessed with getting DNA out of old stuff was I read this paper during my PhD. This is a paper that came out in 2006. And what they did here was in the early days of next generation sequencing, they did pyrosequencing on woolly mammoth, and they recovered 13 million base pairs of mammoth DNA. And what this paper also revealed were some of the techniques involved and some of the limitations and how much DNA is off target when you're working with this old stuff. So less than 50% of the DNA was actually mammoth DNA. The rest is this exogenous DNA, this off-target stuff that's environmental DNA. And how can we detect, they were comparing the mammoth DNA to the African elephant. And we now come up with methods to detect which DNA in your samples is your on-target DNA. They also make the claim that this is unleashing the field of paleogenomics, which I really do think it did. While I didn't know the authors at the time, I just thought this is amazing, innovative, and it's going to change everything. The first author ended up contacting me later to collaborate, and so I'll talk about that later. Since this paper, they've recovered DNA from all sorts of extinct species. Here you see the woolly rhino, extinct sloths, the glyptodon, and then the thylacine or the Tasmanian tiger. And all of these papers have contributed to sort of driving these methods forward. And now we have really nice optimized techniques for getting DNA out of old stuff. And some of the recent papers, I think the specimens are as old as a million years. And so it's really a game changer. For me, though, I don't work with ancient stuff. I work with what I call historic, so recently dead stuff. And so when I'm talking about recently dead stuff, this is stuff from museum collections, which typically date back 100 to 200 years. And this is what it looks like in one of these mammal collections. You have drawers and drawers of these, sometimes we call them little trophies of rats. And so it's these skin specimens that are preserved. You have little vials filled with bones. But nearly 4 million mammal specimens exist in Canadian and United States natural history collections alone, with specimens dating as far back as the mid-1800s. So this provides sort of a, we call it a vault of all these potential projects. And I'm not listing all the projects that, we could, all the different sorts of studies you could do with collections like this, but I'll just kind of highlight some of the stuff that I've done. For example, we have found cryptic species that otherwise would have gone unrecognized because they look like a taxa that's already been described. For resolving phylogenies, this is a big one for me because sometimes we can't get back to those sites where specimens once occurred. Sometimes type localities, for example, sometimes the habitat's simply not there anymore, but they're represented in the museum collections. And so it's really helped to fill in missing taxa and missing locations for some of my data. And more recently, I've started using these museum collections for pathogen screening for both DNA and RNA viruses. And then, of course, one of the things that I kind of want to highlight that we're doing now is looking at how these populations have changed over time with a local project we're doing here in Chicago. But there are limitations to working with this type of DNA. The primary one is it's degraded. And so you end up with really short DNA fragments from these museum specimens. Also base misincorporation. You also get low yield. So the concentration of the DNA you do end up extracting is really low. And then you might be excited when you get like really high yield, and then you discover later that it's because there's a bunch of bacteria or other contaminants in there. But now is the moment to sequence stuff that is degraded. The Illumina sequencing technologies actually favor short fragments. And so fragment length of DNA is a major driver for error rate on this platform. And so read quality decreases for fragments that are over 500 base pairs. So smaller fragments are better. And that's what we end up getting. And so as a good scientist, I wanted to test it for myself. A group of us at the Smithsonian that were kind of all learning from each other. We were all working on postdocs at the same time. We were all about to put together these big museum requests for samples. And the curators obviously don't want you to damage all of their specimens. And they want the most bang for their buck. And so they wanted us to do a pilot test to see if we could get DNA out of old stuff. So our sampling ranged from like 1898 to 1968. And then we looked at a variety of different tissue types. Some are more invasive than others. And so we have bone. Sometimes a museum will let you destructively sample bone if the museum sample is already compromised. Claw and skin. Skin samples are a very common one. And finally, this stuff we call crusties, which are osteocrust or the dried skeletal material that's still attached to the bone. And those are typically things that we would loan or ask or request from a museum. And then the projects we were working on were skunks, rodents. I was working on that big African rodent project at that time, and then marsupials. So these range in size of the animal, and they also differ in preservation type. So I was working on like these little pygmy gerbils that probably dried really quickly in the field conditions. Whereas skunks and the marsupials, sometimes they would even tan these skins. And so they might have had chemicals. And so what we found is that you could get DNA out of just about anything, right? But the concentration as predicted was low, mostly around 10 nanograms per microliter. And we also found that the DNA fragment size as predicted was low, somewhere in the neighborhood of 200 base pairs. And then we did an experiment looking at copy number for a mitochondrial gene and a nuclear gene. And we found that copy number, there was like lots of template available for sure for the mitochondrial genome. And then lots of copies, lots of template for the nuclear marker as well. And one thing we found after kind of taking a closer look is that those skin samples and even nail samples from the tan specimens were problematic. We could still get DNA out of them, but they didn't perform as well. People a lot of time ask me about age. Did you see an effective age of the sample? And we compared this to copy number. And I really don't think that it's about the age of the specimen. I think it's about how quickly that specimen was preserved in the field. We found that specimens that were prepared in the tropics, for example, where if you prepared a skin, it would take longer for that skin to dry. And so, whereas in more arid regions, the museum specimens would dry really quickly. And what we also discovered from this is that the claw samples worked really well. And so we ended up kind of looking at a couple of different methods for getting this fine powder out of the claws. And you can use a drill, a Dremel tool, or a lot of the paleontologists use a dental tool. And what's really nice about it is that you can clean the outside of the specimen before you do the drilling. So it gets rid of a lot of that contamination up front. And so you just end up with this small amount of powder, but it has plenty of DNA in it. And so we really, I personally have a preference for if the claw is big enough to drill right through it. And then ultimately, most of these historic tissue samples, we can get plenty of DNA for next generation sequencing. So our loans were approved. And this led us to this project where we would use some of these DNA samples to look at the systematics of spotted skunks. So you probably know the spotted skunk, I hope. It's a skunk about the size of a squirrel. We don't have them here in Chicagoland. You just have the striped skunk. But I'll show you the distribution of these. They're throughout the US and into Central America. But they probably only weigh about 2 pounds. They're a really, really small animal. And one would think that this is like a carnivore that occurs from the United States all the way into Central America, that we would have those solved by now. That most of the mammals, we would have a good handle on how many species there are. But this isn't the case for this animal. And what's even more surprising, it's a pretty charismatic little animal. Its threat display is to do one of these handstands. And I won't bore you with the historical taxonomy. I just want to point out that if we look all the way back from Liechtenstein in 1838 up until 2017, between 2 and 14 species of these spotted skunks have been recognized. And more recently, they've kind of honed in on about 4 species. And this is where they occur. So we have the western spotted skunk, Spilogale gracilis, the eastern spotted skunk, Putorius, the pygmy spotted skunk in Mexico, and then Spilogale angustifrons. And so we wanted to look across the range. Oh, I also want to point out that you, especially if you teach intro bio, might recognize the spotted skunk as an example of prezygotic isolating mechanism. So it's an example of temporal isolation. And so the Westerns breed earlier in the year than the Easterns. And where they come into, and this is important, where they come into areas, there's like a few places, like especially in Texas, where both species overlap. And so they breed at different times of the year. But what's also interesting is that those Westerns will undergo delayed implantation, such that both the Eastern and the Western have their young in the spring. And so that's a pretty nice example. One of our earlier studies, we just looked at the Western on its own and using a mitochondrial marker. And it was neat. We could see that across its range, there were 3 pretty divergent clades, and that Pleistocene refugia played a role in this phylogenetic pattern. And there were refugias, including in the Sonora Desert, that resulted in this unique Arizona clade. Then a master's student went on to look at the Eastern group. And she found that there were 2 pretty divergent clades here. And that these groups, these two different clades could be recognized as separate species just based on like the genetic divergence compared to other skunk species. And so from that, we had a good handle on the genetic variation in the easterns and the westerns, but to make a case for how many species there are, we needed to look at molecular markers. And we also wanted to include the entire range and locations where previous species had been described and or subspecies that had been synonymized. And so here's our sampling effort. And you can see in circles, there are the modern samples. And then with the triangles, you can see the historic samples. So we really, and with the skunk work, you can probably predict, we get a lot of our modern samples from roadkill. So these guys, like during breeding season, they just aren't even thinking straight. And so they'll cross the roads trying to find a mate. And so we get a lot of roadkill skunks. And I don't want to say the epicenter for skunks is in Texas. It's just where we have the most people collecting skunks. And so we had pretty good sampling. And so we had representatives from what we thought would be the major clades, and we even got a lot of the stuff from Central America from these historical specimens. And so the mitochondrial genome for mammals is around 16, 17,000 base pairs. And so we started with a mitochondrial phylogeny, and you can see here we recognize 7 species. Leucoperia, Angustifrons, Gracilis interrupta, Putorius, Yucatanensis, and the pygmy spotted skunk is always like really divergent, Spilogale pygmaea. And the other thing you're going to notice is we call this the mitochondrial sonora clade. This is a clade we pick up in the mitochondrial data set only and not the nuclear data set. I think what was most exciting about this phylogeny is the Yucatanensis. So we thought that everything from the Yucatan would be Spilogale angustifrons, and it turns out that it is a separate lineage that is not even sister to angustifrons. So there's a unique species in the Yucatan Peninsula. I also did UCEs. I really like when we're working with these historic specimens to use baits. And so I just used the tetrapod bait set for UCEs. And this is a 60% matrix with almost 4,000 UCE loci. So these are 4,000 nuclear loci. And we get the same pattern. You're not going to see the Sonoran clade in there. Yes.
So what are UCE loci?
Okay, they're ultra conserved elements is what the acronym stands for. And they are conserved parts of the genome, but that are found, like I'm using a tetrapod bait set that was developed probably from chickens on a mammal. So the baits themselves are really conserved, but the flanking regions on them are quite variable. And so when we do the trimming and stuff, we can do it in such a way that we're using most of the variable regions, and that's what we're picking up here. Because a lot of times people are using these UCEs for stuff that is above the genus level. But I was able to use it on this, within a single genus. The same patterns here, 7 species that are well supported, and it's like the first time we're getting some of these taxa in the tree at all, including Spilogale angustifrons, which was previously not included in any sort of data set. So it's exciting, and we did a dated phylogeny here. You can see that we can still call one the Western lineage and an Eastern lineage, and the timing of the divergence of these corresponds to events during the Pleistocene. Another thing that I thought was pretty interesting, so I did a spatial diffusion analysis where I'm basically looking at where these lineages are distributed through time. And what you'll see, in the early period, 3.8 million years ago, there seems to be an origin in Mexico, and then it radiates out into Texas, and then radiation further into the eastern and western groups. But Van Gelder already knew that and made that prediction in 1959. And so I just wanted to give him credit, showing that after the Wisconsin movement, how these lineages distributed through space. Okay, so then that left us with these 7 taxa. And you can see where the type localities are and all of that. And this group is actually, the spotted skunks are of conservation concern. Their populations have really declined. And so this has been really useful for folks making management decisions while the maps and the distributions are pretty crude. They're now able to manage these as separate taxonomic units. And so lots of people are on the ground now trying to figure out the ecology of these, behaviors, and so on. And it was really fun because it was, I think, because they do the handstand, the media like really latched onto this. And it was picked up by like 120 news outlets across the world. And we're like, get contacted daily. I even got like weekly, somebody will contact me and be like, so I had this skunk. So it's really turned into kind of a fun paper for us. But I want to switch to talking about a project that we shifted to during the pandemic. And so my partner in crime and I were at home finishing the spotted skunk paper. And we wanted to work on a project where we could keep students engaged, keep students involved, even if it's remotely. But if we can find a way to get them into the lab a little bit, it would be great. And so we came up with this project, and we call it Team Cove Hunter. But basically, we were going to screen the Field Museum's cryo frozen tissue collection for coronaviruses. And the Fields Museum gave us a pool of money to do it. And there's Adam, who was kind of the start of this whole thing. And then Hendrik had contacted me. He was interested, so he's that guy from the Mammoth paper. And he contacted me about a couple of projects. I've got to get this virus stuff going. This is hot right now. And he's like, I've got a way to get RNA out of formalin fixed specimens. And so if you guys have really old rhinolophus bats from, that you want to look at, let's get this going. And so all of a sudden it was like this big group of people that wanted to move this forward. And so the collectors at the Fields Museum include the curators there and mammals, and then me and Adam. And then of course, me and Hendrik were going to drive the lab stuff. He's going to do the ancient stuff. And then I would work with the students on the modern stuff. And then we had too many samples. We had way too many. You can't screen all of the Field Museum stuff. And we had a small budget, and then it's just us. This is like a very small team. And especially during the pandemic, like Hendrik's lab was even doing like diagnostic tests and stuff for COVID. And so we needed a really refined number, like how many should we screen? Which tax should we screen? What tissues should we screen? And so on. And so that's where we brought the modelers in. And I realized this is like extremely like sex bias here. I'm working on it, but they're all really great folks. And so the modelers helped us come up with the numbers. And so I also received money from the Institute for International Education. This is like the same funding agency that funds Fulbright. They said, they sent out a call to all the previous Fulbright alum, and they were like, if you're working on a coronavirus project, submit a proposal and we're gonna fund 4 of them. And so they funded us to carry on and do this with the students. And so that's been a lot of fun. So the big thing is, in the early days of the pandemic, that the pangolin and rhinolophid bats were implicated as the reservoir host for SARS-CoV-2. And then as it progressed, as there was more human-to-human contact, then they're spread to captive animals, like the zoo animals and mink farms. But we still didn't know where it's naturally occurring, besides the rhinolophus species that was implicated. And the Field Museum bat collection, though, has tons of stuff to screen. Here, just an example of some of the samples I sent off to Hendrik, these really old rhinolophid bats. And so the modelers looked at the amino acids in the ACE2 receptors for mammals, and they looked at whether or not betacoronavirus had been documented before in a host. And they came up with a predictive model for us. And so we narrowed it down to 1,500 specimens that we would screen. So we're not finished yet, but I'll present some of the student results. We go to this conference, ILSAMP, and they've been presenting it there and elsewhere. And what we found is that we do pick up alpha coronaviruses and beta coronaviruses in these samples. And we're picking up definite geographic signatures. So if we get coronavirus positive samples from Angola, for example, they'll cluster with a unique Southern African clade. And so here, for example, there is a beta coronavirus isolated from a Rhinolophus species from Angola that was in the Field Museum collection. And here's the entire tree in green for beta coronaviruses. And our sample clusters in sort of a sister group to human SARS-CoV-2. So we need to look beyond, Asia when we're thinking about beta coronaviruses, that in Southern Africa and Sub-Saharan Africa, it's circulating, beta coronaviruses are circulating as well. And so to be continued on that, I'm hoping to have more data soon. The students are also, for all these 1,500 samples we're screening for coronaviruses, they're also looking at, when we do pick up a positive individual, they're looking at the ACE2 receptor. So they're doing DNA work as well. Okay, so then I really want to talk about this project too. This is work we recently started in the Chicago area. And again, my whole mission is to get undergraduate students really involved in research, and how can I make these projects easy for them to do. I don't know if we made this one easy for them, but it is something that's really close to Chicago State University. And so Adam was funded on this one. This is we call it the Small Mammal and Pathogen Diversity in Southside Parks in Chicago, with implications for human health and well-being. So he's really trying to take this One Health approach where you're looking at human health, environmental health, and animal health. And it was funded by the Walder Foundation, and our partners include folks from the Fields Museum, the Forest Preserves, Wheaton College, Chicago Parks, Lincoln Park, University of Illinois, and also me at Chicago State University. And so we came up with this crazy acronym. We call it the CAPTURE Project, but it's the Calumet Animal Pathogen Training Urban Research Experience. Here's a picture of, if you haven't been down to Big Marsh yet, it's right very near Chicago State University. Ford has a nice environmental center there, and the Fields Museum helped put the diorama in there, and it's got all this educational stuff in there as well. Really cool bike trails, and it's kind of near Pullman, so you can do like a little loop over there. But the idea was, ever since I was at Angelo State University and we were doing bat survey work, my advisor at the time had like a 15 years, probably 20-year long survey of bats in Big Bend National Park. And for a lot of these kind of data sets, the pattern really doesn't become revealed until after about 10 years. And so me and Adam started doing the math, and we're like, we need to get a study site going now. And so we decided to do it here very near Chicago State University, so it'd just be easier for the students to go. And it includes these sites, Big Marsh, Eggers Grove, Bobian Woods, Miriam Burns. We actually eliminated the Dan Ryan. And we're doing small mammal trapping, live trapping with these Sherman traps. This is a capture, mark, recapture survey. So we're keeping the animals alive. We're doing tick drags, and we're also doing pathogen screening. And so the questions we're trying to ask are just general stuff about mammal diversity. Is pretty low here, but looking at how they differ across the sites. The second one deals with tick populations. And so we're looking at infestation rates based on the different land uses we're seeing at these sites. And then we're looking at pathogens and how those differ across sites and species. And then finally, there's like a social science component to this, where we're involving the community. Now, we just finished our pilot year. And so in December, we closed the project down, and we're ramping up for the next season, which is gonna start in April. And then it'll run continuously through the fall. And so the pathogen of interest are Lyme disease, which is transmitted by the deer mouse, Borrelia burgdorferi. We're interested in Rocky Mountain spotted fever. And then we're also interested in raccoon roundworm. So you saw Wheaton College was one of our collaborators. Kristen Page is there, probably one of the few raccoon roundworm experts. And so we're going to be collecting, we already are collecting, we mark the animals with a little ear tag, and we take a blood sample, and we take an ear clip, and we take all of the ectoparasites. And then any mortalities, it's really nice. You do end up with mortalities for these sorts of studies, but they all go into the Field Museum collection. And what's super surprising is the Field Museum collection is like a world-renowned collection, but it doesn't have very many specimens from Illinois or this region. And I'm like, you guys, but the international, like Madagascar stuff, they're like #1, and a lot of stuff from Africa and so on, and South America. And so we're building up this collection. There is some material we're going to look at that's from about 100 years ago, but I mean, there'll be like 5 of these deer mice from this region. So, and it's a really student-focused project, and I'm bringing this up because it's not just CSU students. Students from Wheaton College are going to participate, and we're just trying to get a lot of local participation. So if you know any students that want to get field experience, we have volunteer positions that you can run through the Fields Museum, but we also have internships, paid internships. And so I'll shoot those to Windsor once we get those advertised, and he can advertise them more broadly here. But just people that are interested in ecology, and we need a lot of help in the field, but then we'll need a lot of help in the lab too. And so Zoe managed it this season, and she did a fantastic job as the field sites coordinator. And then we've recently hired a master's student, a new student to Chicago State University that's going to take over for it. And then of course there's going to be a lot of CSU students that come in and out of the project. Okay, so that leaves me with, so yes, tell your students. These will be advertised for both the volunteer positions and the internships will be advertised through the Field Museum. So what's next for me is this. So I was just awarded an NSF grant to carry on with some stuff that I started during my postdoc. So my second postdoc at the Smithsonian was looking at mammalian pox viruses. And the reason I got involved in this is because if you look at human smallpox, I don't think this is the tree I meant to include. Let's see if I can find it here. Oh, I see it. Okay, so human smallpox, variola virus is up here. And when you look at its closest relatives, it is this thing called camelpox and gerbilpox. Okay, so there's seemingly an arid origin for human smallpox. And while I was at the Smithsonian, they have a huge series of rodents, just mammals in general, from sub-Saharan Africa. And a lot of this collection was done from the 1960s into the 1970s, and they would just send folks from the Smithsonian out into the field. And the point of it was disease surveillance, and they were really interested in human smallpox. And so this is a bunch of dots on a map showing where the Smithsonian went in some of these screening efforts. And the other really crazy part to this story is that the gerbil for which gerbilpox was isolated from only a single individual from Benin in 1968. But they have a very large series at the Smithsonian of other small mammals from the same region, from the same period. This is during a time period when there were known human smallpox outbreaks in the region. And so I just kind of focused in on, here's the area where, you can't screen everything, but I'm working with Hendrik Pointer on this project as well. And he's got really clever ways of like, if you've got a lot of samples, we can do this to save some money. And then once we get a positive, then we'll look at a bunch of individuals. And so that's what we're going to do. We're going to focus on this, like, can we detect gerbil pox and other taxa. We're also going to be looking at monkeypox. And then there is a newly described lineage of pox viruses isolated from human and some small mammals in Fairbanks, Alaska. This outbreak only happened recently. And it falls out in a super weird part of the tree. And so we'll be screening the Fairbanks collection to see how far back in time we can isolate Alaska pox. So what else have I got? I think that's it.
So we have time for questions.
Yes.
You said that there were the old collections from 1898 to 1966, something like that. And I was curious if there were like collection notes for when the samples were collected, because you were talking about how ones in different areas had different techniques that they were preserved in.
Yeah, I did go back to field notes for this one because I was really, so the marsupial samples weren't working well. And I was like, well, maybe if we go back to the field notes, we can see if they were preserving them with arsenic or something like that. And so we were able to track down some of the preservation techniques from field notes, but not all. It's actually pretty uncommon for people to describe how stuff was preserved. I wish, that's a lesson in just take as much detail as you can in your field notes. You don't know how people are going to use it later. But that's a good point. Yeah, some of these collectors did a really good job of recording everything.
I have a couple of questions. So one of them is with the viral work you're doing. I just think it's incredibly fascinating, and I wonder how much with museum collections, researchers often go when they're collecting in an area and they have their objectives, and then 5 years later something happens. There's a major environmental change in the area, and then you can get modern collections, or there's other collections that happened afterward. But it almost makes you wonder, beyond looking at where the virus is, looking at how they're evolving, especially as things are happening, invasive species get introduced, forests are cut, livestock species are being moved in. It makes me wonder how much you can use these collections of rare mammals to look at the evolution of these viruses that some of them are potential zoonotic agents for humans.
And some of the collectors that I listed there, like Steve Goodman's in Madagascar all the time, and he's collecting all the time. And then Larry was constantly going to the Philippines every year. And so some of these collections, yes, you could look at transects through time, and others maybe not. But it's really helped us identify how we can use this collection in the future. And one of the big problems we had up front. So Adam came before I did, but we've only been at the field for a pretty short period of time. And he didn't have a good handle on what is the quality of all this stuff. And so this project, we really did deep dives into the cryo stuff, and we were able to record we didn't even know if it was in a certain type of buffer, if it had viable RNA still in it. So it's kind of changed how we think about how we're going to collect stuff in the future and how we can use the existing material that's there.
And another question I had was with the spotted skunk project, were you able to include extinct populations in from the historical records?
That's a good question. I don't know if I don't know. I'll ask Adam. I think that it's very possible that our sampling included extinct populations.
Yeah, because I wonder, the other thing, my mind goes to when I see your research is, humans are having this huge impact on the environment. Many species are going extinct, but also populations.
Yeah.
And sometimes those populations are locally adapted, and there's aspects of their genome that have evolved in that area, and then they go extinct, and that's lost from the species. And kind of finding ways to understand what was lost.
Yeah, exactly.
And even if a species exists in these populations, what did we lose? How did they work and
Yeah.
manage to survive in these
That
areas?
would be worth looking at. It's been kind of neat with like the publicity this has gotten within the mammal community. It's resulted in a lot of people contacting us. Like, we know that Yucatan population, Yucatanensis, is doing well because some vets have samples that they were screening for rabies. And so they're like, no, it seems like it's a good population. So we're now starting to like hear how different, like some populations in California, they wanted to know what the common name was and so on, but could tell us something about the population too. So there's actually a lot of folks working on the spotted skunks, and it's been a fun project.
There are other questions, yeah.
Oh,
Go ahead.
so I think Chicago is flying squirrels, right?
Flying squirrels?
Yeah.
Yes.
So can you capture them? Can you look at
Yeah.
flying squirrels?
Yeah. They even, like one of the curators was telling us, they come to the bird feeders. But yeah, there are specific methods for trapping those. I do know they like coming to bacon. Bacon. Yeah, I don't know anyone that's, I don't personally know anyone that's working on them, but yeah, they're really, I think not difficult to trap.
And have you looked at them at all?
Yes.
I mean, not physically, but like their DNA.
Oh, no. But there's a nice series of them at the Fields Museum.
From my own experience at the bench, RNA is much harder to work with than DNA.
I'm not going back to that. I hate
So
RNA.
could we talk about some of the differences about extracting nucleic acids from old samples for DNA versus RNA?
Okay, so the stuff that I was screening with the CSU students, we were only working with stuff that we thought was flash frozen in the field. So then it goes from the field to the cryo facility. And so then the cold chain really wasn't broken. I mean, it's a pain even when you're like trying to do the extraction and dealing with something that degrades so rapidly. But the historical stuff, I usually don't mess around with formalin-fixed stuff. Like you can get DNA out of it, and it's just like bioinformatically a little bit messier to deal with on the back end. But Hendrik, he's a molecular biologist, so he has this explanation for why formalin actually preserves RNA, and that he can get pretty good RNA out of formalin-fixed stuff. So for the old stuff, 1800s, he was only working with formalin-fixed back colons. So we did a kind of a tropism study. One of the students did this where we took tissue samples that we knew were flash frozen on a recent trip to Angola, and we took as many tissue types as possible, and they were all flash frozen within 2 minutes or something like that. And so she could look, okay, here's a positive animal. Let's look across the entire digestive tract and other tissue types as well, and see where we can find coronavirus. And so we determined that the colon is like the best place if you have colon samples. But then we could detect it in other tissue types, just fewer reads. But I hate working with RNA. I don't want to work. So pox viruses are DNA. So you can see this transition is going to be much more pleasant. But I wanted to design a project that the students could do too. So what we would do is we would do extract the RNA. We did a kit where called prep, where you could extract the RNA and the DNA simultaneously. And so I had the students working a lot with the DNA at first, but then with the RNA, I wanted to do a PCR-based method, and they could run it out on a gel. And so we did like a nested PCR and get enough copies in there to where they could visualize it on a gel if it was positive. Now I know there's a lot of error in this, and we were probably getting a lot of false negatives, but it was the best we could do, I think, given all of the situation. But yeah, I hate RNA. No offense, RNA. Yes.
I know you primarily work with DNA, but do you ever see a situation in which these methods could be applied to searching collections for old world viruses such as Marburg?
Oh yeah. Yeah. And if you're, yeah, so it should be publicly available. You should be able to search the Field Museum Mammal Database. And I think that you can even see what tissues are available. It's like a little bit sketchy in terms of tissue type. But you can always reach out to Adam's gonna kill me. Reach out to the collections manager. I'll give you a list. But yeah, I mean, yes, definitely. There is tissue preserved in a way that the RNA would remain intact for RNA viruses. Yeah. And that's what we're always thinking about. So when we go, like you saw those places we go to, this is how we were trained. You go to a site and you're gonna get them, if you're gonna kill an animal, you get the most amount of information you can possibly get. And so if that meant taking, 12 tissue vials, it meant taking 12 tissue vials. But then now there's a lot of people using whole specimens for CT scanning. So we would have a list of like, okay, here's the taxa we need to get as many tissues. Once we max out on that, we're gonna get whole bodies so that people can use it for CT scanning. And yeah, so we try and do this kind of like holistic sampling approach. Yes.
So now that, back to this started skunks, now that you know the species boundaries based on molecular
Yeah.
data, now how about the morphological?
Yeah.
Is there a gradational still? Or is it
We want a master student to do this.
Oh.
We want a master student to do this. If you know anyone, we would love, that's the next thing. So Van Gelder in the 1950s, the one that did that nice map of how they distributed, he looked at morphology, but it was like, it was a little difficult to sort everything out. And maybe if we put it into this context with 7 species, that a student would have more luck. So we're hoping. They tried to use spotting pattern and cranial measurements, and it never really turned out to be super useful. So yeah, we want a master student to do this. Yes.
Going back to the spotted skunk, you have that clade that's in the Yucatan,
Mhm.
and then you have its sister to a clade that's in the temperate eastern North America, right?
Yeah. I'll do this one.
So it's okay, yeah, sister to the yellow and
Yeah.
so the plains and like southeast.
Yeah.
So that's kind of a weird distribution, right? And is that just because like that intervening part of Mexico dried out in the interim?
I don't know. It could be. Yeah. It would be nice to do maybe some niche modeling.
Yeah.
on this.
Can they swim well?
Yeah. So that's what, if you look at my, according to my spatial diffusion analysis, they swam.
I saw that, I'm like, that can't be literal.
It drifted.
Any other questions?
Nice to meet y'all. I'm just down the street.
Yeah, so, okay, so we have just a couple things more. So one as a little thank you for coming to attend. You have a certificate which comes with a $100 gift card from
know that.
Amazon. Yeah, so that's for you. And
Thank you.
I also need to thank the seminar committee, Carolyn Martino and Jason Mistransky, who help organize these things. So I get to talk and introduce people, but the seminar committee does the work. And then we have a bunch of treats that are in here. We planned on them being outside because there's posters outside. So you're a smart group. I hope you figure it out. But there's here are posters outside. Interact with the posters, and then I hope you have a good time and take what you need. Thank you. Thank you.